Thank you very much. It's a great honor to be here, and it's my pleasure to introduce our Singapore longitudinal birth cohort, as well as I'm going to present a subset of studies derived from this study. Especially, I'm going to discuss some of the findings we have that related to transmission of maternal mental health to the offspring, based on the evidence derived from pediatric imaging as well as genetic data. As we all know, prevalence rate of mental illness increases dramatically in early life, indicating a need for understanding of developmental origin of mental illness. We are nowhere close to understanding environmental factors and biological processes that shape brain development in early life, which may lead to mental illness later in life. We do not know how to predict which child will develop mental illness and what form the mental illness will take and how to prevent such fate. More broadly, we do not know how to maximize the potential of individuals since young. In 2009, in Singapore, we started a longitudinal birth cohort study called Growing Up in Singapore Towards Healthy Outcomes, GUSTO, at two major hospitals in Singapore, including Women and Children Hospital, KKH, National University Hospital, NUH. We aim to look for answers to these three challenging questions by following up the family since pregnancy. In GUSTO, environmental factors include complications during pregnancy, maternal mood, nutrition, family factors such as socioeconomic status, family support at each time point since pregnancy, child nutrition, parenting at each time point after birth. As for outcome measures, child cognition and metabolic measures, as well as epigenetic data, were assessed since birth onwards, and body and brain imaging were acquired at multiple time points. So just give you a brief overview what we measure in cognition and brain imaging. In the first 24 hours life, we conduct a facial imitation task for prediction of social skills of the child. Brain imaging was acquired in the first 2 weeks of life for diffusion tensor imaging as well as T1 and T2 structural imaging. At 6 months of age, we adding on resting state data. So in addition, at 6 months and 18 months of age, we employ event-related potential, eye tracking, as well as video recording to assess child executive function, attention, memory, as well as mother-child interaction. From 2 years onwards, we conduct visits once a year. After 2 years of age, we start to measure a child executive function, memory, emotion, and theory of mind at each time point. Bailey scale of infant and toddler development was conducted in 2 years for assess a global cognition as well as motor skills of the child. Child behavior checklist was conducted at every time point since 2 years onwards, and child psychopathology was conducted at 4 years and 8 years onwards. As academic outcomes, we also measure language, numeracy skills of the child, as well as intelligence, IQ, at 4 years of time. And each sketch and adulthood attachment were assessed at 3 years as well as 5 years for mother-child interaction. And brain imaging was restarted at 4 years age. We repeat the scans every 1.5 years. To just give you an idea what brain imaging look like at birth as well as at 6 months of age, here we show the DTI and T2 atlas generated from our data based on a probabilistic atlas estimation method developed by our group. The first 2 rows shows a color map and fractional anisotropy maps derived from diffusion tensor imaging, and last row shows the structural T2 images. And you can see the brain grows dramatically in terms of overall size in the first 6 months of life. The white matter tracts become more coherent. Certainly, there's a challenge of imaging 6-month-old brain using a conventional MR structured T1 and T2 images in a limited scanning time. Given all this deep phenotyping data, what can we explore? As mentioned earlier, one of our interests in Gusto is to look for environmental factors that could shape brain development in early life. A substantial body of literature suggests that prenatal maternal depression increases risk for psychopathology of the upstream in adolescence and adulthood. Question to us is that whether the neonatal brain remember the fetal exposure to maternal depression. In other words, does the brain of neonates born to the mother with depression look like the brain of depressed adults, especially in the cortical limbic circuitry? Based on the neonatal imaging data, we show that the neonates born to a higher prenatal maternal symptoms had lower fractional endostropy as well as axial diffusivity values of right amygdala compared to those who born to the mother with lower prenatal maternal depression. And based on a 6 months resting state imaging data, we also showed the greater prenatal maternal depressive symptoms are associated with a greater amygdala functional connectivity with the structure in the sensory, memory, as well as emotion regulation networks. These findings suggest that neural substrates associated with increased vulnerability for depression are established as a function of intrauterine maternal depression infancies. Does prenatal maternal depression have a long-term impact on brain development beyond the course of gestation? Well, there are substantial inferences from postnatal environment. Our data acquired at 4 years of age showed that greater maternal depressive symptoms were associated with a greater right amygdala volume in girls, but not in boys. The gender specific was not emphasized in the neonatal stage. And this is true even after controlling for postnatal maternal depressive symptoms and other environmental factors such as socioeconomic status. And this finding is also in line with what we discover in the 7 years of old children. Claudia Bass, as well as colleagues, showed that greater prenatal maternal cortisol levels predicts a larger right amygdala volumes in 7 years girls, but not in boys. Furthermore, resting state function MR data also show a link between prenatal maternal depressive symptoms and amygdala functional connectivity in girls, but not in boys at 4 years of age. Such association happened in the same structure, such as orbital frontal cortex, superior temporal gyrus, as what we see in the infant data. However, the association direction is opposite to what we see for the infants. It remains unclear when is the turning point and what drives such changes. So nevertheless, we show the fetal exposure to maternal depressive symptoms has a long-term impact to brain beyond the course of gestation. At least in our sample, sex-specific inferences occurs after birth. So what about our genes? Do they play any role in such relationship? As we know, depression is a polygenic trait. Individual SNPs explain a very little variance of depression. So in our study, instead of looking at individual SNPs, we select a set of SNPs that are at a certain level of risk for depression based on existing GWAS depression study. We then generate polygenic risk scores to indicate the individual genetic risk for depression. So the polygenic risk basically modulates association between prenatal maternal depressive symptoms and right amygdala volumes of neonates. greater prenatal maternal depressive symptoms were associated with a greater right amygdala volume in neonates with a higher genetic risk. We see the opposite direction for the neonates in the lower genetic risk group. We then took the genes that most contribute to such modulation effects and performed a gene network analysis. We discovered that genes highlighted in yellow that forms a glutamine receptor activity as key genetic networks that regulates the relationship between prenatal maternal depression and neonatal amygdala. And this finding can be replicated from clinical sample acquired at University of California, Irvine. The same genetic network, glutamine receptor activity, was identified in this clinical sample. So as we know, patients with major depressive symptoms exhibit abnormal glutamate concentration in the amygdala. And certain depression medication blocks stress-induced increase in glutamine release in the amygdala and modulates the behavior response to stress. So our finding hence suggests that such genetic modulation may explain in part the variation in offspring phenotypic outcomes associated with maternal depressive traits. As neonatal brain remembers exposure to maternal environment during the course of gestation, to what degree and up to what age can neonatal brain predict child behavior later on? In our study, instead of building a classifier that links the neonatal brain with child behavior measures later in life, what we asked was whether the neonatal brain structurally wire in the same way across individuals. If not, does the variation of neonatal brain organization explain future child behavior? For this, we basically construct a brain structure network based on the neonatal diffusion tensor imaging data. We then employ a supervised classifier approach and discover 4 different types of brain structure organization in neonates. When we look at the internalizing and externalizing behavior scores assessed using child behavior checklist at 2 years and 4 years of age in these 4 groups, we found that children in group 2 had the best behaviors, and the children in group 4 had the worst, while the children in group 1 and 3 shows intermediate level of behavior scores at age of 2 and 4. We then examined the link of child behavior measures with structural network measures that were used to classify neonates into different groups. We see again that right amygdala structural organization at birth was related to internalizing and externalizing skills of child at 2 years of time, 2 years of age. And such a relationship would become weaker at 4 years of age due to a potential long-term impact of postnatal environment. And this phenomena remains the same when removing the potential outliers circled in red. Therefore, the neural organization established during fetal development could to some extent predict individual differences in behavior profile in early childhood. In summary, the longitudinal birth cohort study provides a great potential for understanding environmental factors as well as the biological process that shape brain development, which may hence predict child behavior profile later on. Today, I don't have enough time to cover the other 2 challenging questions, and you can visit our website and to look for existing publications. Finally, I'd like to thank all my lab members and valuable collaborators at Gusto that I will not be able to list down here for their contribution to this work. I also like to thank a funding agency for supporting this work in the past 10 years. Thank you for your attention. So I turn to Mac to